Hello everyone, thank you for joining us today. My name is Fatima Jama and I'm the Assistant Coordinator for the International Parkinson Disease Genomics Consortium Africa, and I'll be moderating this session. So this webinar is part of an ongoing series of webinars called Inspiring Research in Neuroscience in Africa. And today we're very fortunate to have Professor Racette joining us from the Washington University in St. Louis. So before I pass it on to Professor Racette, I'll give a brief introduction. So Dr. Racette is the Robert Allan Finke Professor and Executive Vice Chairman of Neurology at Washington University School of Medicine in St. Louis, and an Honorary Professor of Public Health at the University of Witwatersrand in Johannesburg, South Africa. He is a graduate of Princeton University, Northwestern University School of Medicine, and completed his neurology residency and fellowship in movement disorders at Washington University School of Medicine. His research focuses on manganese neurotoxicity and environmental risk factors for Parkinson disease. His research is supported by National Institutes of Health, Michael J. Fox Foundation, US Department of Defense, American Parkinson Disease Association, and Cure Alzheimer Foundation. He has led global health research projects for over a decade in South Africa and Finland. His work in South Africa focuses on occupational and environmental manganese with an emphasis on vulnerable populations. He has served as a peer reviewer and advisor for numerous medical journals and international regulatory agencies. He has received numerous awards and has authored over 160 peer-reviewed publications. In this talk, Professor Racette will describe the path to his team developing an innovative environmental manganese neurotoxicity research program focusing on Parkinsonism in South Africa. He will focus his presentation on the challenges to developing an environmental manganese cohort in Merton, South Africa, and will present preliminary results from the study. So with that, I'd like to pass it over to Professor Racette. Thank you very much for joining us, Professor, and I look forward to hearing your talk.
Thank you. It's my pleasure to speak to the International Parkinson's Disease Genetic Consortium, and thank you for the invitation. I chose a somewhat provocative and unique title for a talk to a genetics consortium, but hopefully you'll see throughout the talk the strength of the program we've built in South Africa and how the sort of platform for Parkinson's research that we've developed could be leveraged for your genetic consortium. So, and please, Fatima, let me know if for some reason something doesn't advance. So I'm going to talk briefly about sort of my path to this work and give you some background on manganese. So as was mentioned, I'm a movement disorder physician at Washington University, and my interest in Parkinson's began actually with my grandmother, who's pictured on the left, Lena Racette. She developed Parkinson's at age 65. Lived with the disease for over 30 years. And she demonstrates a couple of important risk factors for Parkinson's, namely age and probably Caucasian ancestry, although that's an important area that we don't fully understand. She also lived in a rural area and there's some evidence that may be a risk factor as well. While this talk is not about ancestry and Parkinson's per se, this group's well aware that there are many challenges to investigating underrepresented populations in epidemiology research. And I'm going to highlight some of those challenges along the way. This figure on the right demonstrates my early career research focus, which actually was genetics of Parkinson's disease. And one of my first NIH grants was to study genetics of Parkinson's disease in an Amish community in central Illinois. And on the top, you see an Amish buggy, and on the bottom, you see a pedigree showing multiple extensive inbreeding loops in the community that we were studying. And we cite there's a couple papers that describe some of the work we did, but ultimately, despite our hypothesis that Parkinson's in this community was likely genetic, we found mostly evidence for an environmental etiology. So along the way, through my clinical practice, I had a clinical observation through a Parkinson's patient who came to me. He was a welder, a young guy, and he asked if his Parkinson's disease could be due to his welding exposure. And so I looked into this, and there was clearly literature, at least in the safety data, suggesting that there may be some risk of Parkinson's disease in people exposed to manganese-containing welding rods. And so we published a case control study where we actually tried to investigate this, and we compared a group of welders with Parkinson's disease to a group of a serially ascertained group of Parkinson's patients from our clinic, and demonstrated that they were clinically similar to our to the general Parkinson's population, with the exception of being on average about 17 years younger at age of onset. So this prompted really a deeper dive into understanding manganese, and the literature of manganese toxicity is really the literature of a disease called manganism. And manganism is a disease that was first described in the 1800s with in a plant that produced bleaching powder containing manganese oxide. And although the descriptions of the clinical phenotype of several of the workers who became sick are rudimentary, they largely consist of a description of Parkinsonism. In the early 20th century, the invention of manganese-containing steel led to a dramatic increase in manganese use in industrial settings, and then that, of course, led to the need to mine more manganese. And in the 1950s, Rodier described a large number of workers that were working in a manganese mine that who developed this is actually a Moroccan manganese mine, who developed a Parkinsonian syndrome, somewhat atypical, with relatively rapid onset, characterized by Parkinsonism, dystonia, and cognitive abnormalities. And so these exposures that were described by Rodier were in the order of 1 million micrograms per meter cubed. These are massive exposures, and well above the modern OSHA standard for manganese exposure, the OSHA limit of 5,000 micrograms of manganese per meter cubed. So these studies suggested that manganese toxicity was a distinct disease from what we had observed in our workers that were working in welding trades. So why should we care about manganese? Well, manganese is interesting because it's actually essential. It's an essential element for several human enzymes. So there is actually a recommended daily oral level of manganese that should be contained in the diet. But it's also common occupational exposure for millions of workers worldwide, including welders, smelters, steel workers, and miners. And there are billions of metric tons of manganese emitted in the United States and worldwide. And it's also found in some pesticides, and in some countries, the fuel additive MMT, which is used to increase octane in gasoline, is leading to dramatic increases in manganese through automobile emissions. So I became interested in this, started to pursue this area of research, and after a couple publications that suggested that maybe this is an area that we should be focusing some research in, and we tried to advance grant applications. The welding industry actually undertook pretty dramatic efforts to stifle some of the work that we were doing. On the left, you see one of many subpoenas that I received for some of the underlying data for work we had published in our early years. We had over 20 freedom of information requests from attorneys. And then the welding industry funded multiple studies, all of course demonstrated no risk of Parkinson's disease in welders. They also paid over $10 million to defense consultants to largely undermine some of the work that we had published. So that was the environment that we were faced with when we started to really become interested in manganese exposure and the relationship between manganese exposure and Parkinson's disease. But despite that, we fought through this. We obtained multiple NIH grants, and I show on the upper right a couple investigators in my lab who helped lead some of this work, Susan Criswell on the left and Dr. Susan Nielsen on the right. And this is just a snapshot of some of the key findings that we've had in our manganese welding exposed workers. We've demonstrated that manganese dose-dependent progressive Parkinsonism. In welders, we've demonstrated MRI diffusion abnormalities in the basal ganglia of welders compared to controls. We've demonstrated a linear association between the pallidal index, which is the bright signal intensity in the globus pallidus from manganese deposition, and the Unified Parkinson's Disease Rating Scale, so a clinical measure of Parkinsonism. We've shown reduced F-dopa uptake in welders compared to controls in the caudate in particular. And we showed upregulation of D2 receptors in the caudate and midbrain in welders, and an association between the UPDRS and D2 receptor binding in the midbrain. And finally, in a recent paper, we've demonstrated an association between the UPDRS and VMAT2 binding in the thalamus and the pallidum. So this is just to show that despite those early challenges, we've done a lot of work to demonstrate that manganese-containing welding fumes is associated with a Parkinsonian syndrome that has remarkable similarities to Parkinson's disease in terms of pathophysiology. But the question is that most exposures in the world to manganese are not occupational, most are environmental, and those environmental exposures are substantially lower than occupational exposures. So that brings us to sort of phase 2 of my career, I guess, and this is the beginning of my African studies. So you see in this picture on the left a picture of me back with a lot more hair and a lot fewer years with my colleague Jill Nelson, ironically at the John Snow pub in London, where I was actually at the University of London at the time, taking a 2-week sabbatical and taking a course in advanced epidemiologic analysis. Jill was my lab partner at the time, and we found that we had common interest in that she worked for the National Institute of Occupational Health in Johannesburg for a program that obtained heart and lung autopsies in deceased mine workers in order to facilitate compensation for family members when workers died with illnesses such as silicosis or asbestosis. So I was, after many discussions about this program with Jill, I became very interested in whether the about 1% of workers in South Africa who worked in the manganese mine industry might be willing to give us brains to do neuropathologic evaluations. And after a lot of persuasion, I ended up visiting South Africa for my first time in 2008. And that led to a series of grants where we were able to develop a cohort of about 400 manganese mine workers in the northern Cape province of South Africa to do clinical work to look at Parkinsonism in those workers. And then we were also able to develop a neuropathology a series of neuropathology projects and a brain bank, and now we've obtained over 90 brains from deceased miners in South Africa. And we've been able to do MRIs in those brains, and then the neuropathology work is done at the University of Washington. And so far, the early findings from this study, and we have an R01 that we're working with right now, but the early findings from this study suggest that manganese induces neuroinflammation in the striatum, and our current grants are focused on trying to understand how that is related to neurodegeneration. And you see on the upper left my first encounter with going underground in a manganese mine. Jill and I went underground as my first and last. It's not something that I enjoyed, but it was essential really to understand the experience that these workers go through while working underground. So concurrent with our work in the manganese mines, we were contacted by the South African Department of Environmental Affairs about a potential health concern raised by a community near one of the largest manganese smelters in the world. We developed and implemented a pilot study, and eventually were funded an R01 and an NIHS Victor grant. The smelter is pictured in the top right panel when it was sort of at its peak of maximum polluting. And as you can see in the picture on the bottom right, the community that we're studying is right on the other side of the fence. And this is the child there in the picture next to the minivan is in a daycare that's right across the fence. So the work that I'm going to describe in the next few slides really is a culmination of many years of preliminary work, and then now with multiple NIH grants that are supporting this project. And the primary aims of the grant are to look at the relationship between cumulative and environmental manganese exposure and motor function as measured by the UPDRS and the Groove Peg Board, mood as measured by the Beck Depression Inventory and the State Trait Anxiety Inventory, and then cognitive control, focusing on the go-no-go test and the DigiSpan test, as well as a non-verbal IQ test. Now, it's important to say at this point that we're specifically not studying Parkinson's disease in our studies because of the rarity of Parkinson's disease in epidemiologic terms. So because Parkinson's affects just a few percent of the population, when you're doing studies with 1,000 people or a couple thousand people, you generally don't have enough power to be able to identify these associations. So in order to circumvent that, we study Parkinsonism as a continuum, and there's a long rationale for why that's a valid approach, but largely this has been very effective in our occupational studies and has allowed us to detect dose-response relations and to do very novel neuroimaging studies looking at associations with regions of the brain that are pathophysiologically related to PD. So you see on the right a graph of the manganese concentrations that we're studying. So hopefully you can see my cursor. Our welder studies are up here in the range of about 0.14 milligram of manganese per meter cubed. These studies are well below the OSHA permissible exposure limit. But when it comes to environmental levels, our exposure levels are orders of magnitude lower than the exposures that you see in occupational settings. So the importance of this work lies in the fact that tens of millions of people worldwide are exposed to environmental manganese levels in this range, and the range we're studying is below the EPA recommended exposure limits, and is well below what many international regulatory agencies determine to be a safe level of environmental manganese exposure. So our studies have the potential to inform environmental manganese regulations worldwide. Concurrent with this grant, we have an innovative biomarker discovery grant, and I just mentioned this briefly, where we're collecting saliva from every participant in our study, and we are isolating microRNAs, which are non-coding regulatory RNAs that likely function to regulate gene expression. These microRNAs are being isolated from our research participants, and we're looking for the association between each microRNA level concentration and manganese exposure. And in concurrent with this, we're doing similar experiments in Drosophila in Dr. Yuda Ben-Shahar's lab, and in a human tissue, in an ex vivo neuronal cell model with Anu Kanthasamy at Iowa State. And the idea is that we look for homologies across these different model systems, consistency, I should say, and using that to inform our biomarker discovery. So an important part of this study, of course, is exposure assessment. And on the left is an aerial view of the community we're studying. This is the exposed community. Our low to no exposed community is about 60 kilometers northwest of Johannesburg, of Meyerton, and just in the northern outskirts of Johannesburg. So the Meyerton region, which is what this shows the smelter there's a very large footprint of the smelter on the left. And then the three settlements that we're studying, New Sikelo, Noldik, and Old Sikelo, are labeled accordingly. And then to do this study, we performed fixed air sampling in 3 locations in the community over a 4-year period of time. And then in addition to that, we took measurements from a sampling of homes in the community, and were able to demonstrate that these measurements were strongly correlated. We originally had planned on taking these fixed samplers, these large samplers that are shown here in the middle picture, and putting them in individual research participant yards to get more refined measurements, but it proved to be impractical because of the concerns over theft of the equipment and just general security. So we had to modify that plan. In addition to the fixed sampling, and for every research participant, we were able to calculate an individual level exposure based upon exposure modeling, which I'll show in the next slide. And that's done by geocoding the residents, and then by taking some very detailed home measurements, for example, measurements of the home, distance from the home to the street, so that we can do more refined modeling. And that's what Melanie's doing here in the right picture. So our work in South Africa is based at WITS, as you gather, and really this is a tale of 2 different worlds that we're working in. So WITS is a well-renowned school of public health in South Africa, and really has amazing educational and intellectual resources. But the environments we're studying are actually quite complicated. They're more rural, and we really focus our work in the formal settlements, which for those in the United States or in the UK, these really represent government subsidized homes, and they're a way for us, because these are organized communities, we're able to actually go door to door with our sampling. We actually had a random sampling approach, but we can go door to door to do recruitment because we have a map of the entire community. But one of the things you'll notice is that even in the formal settlements, there are these other structures on the property. So multiple homes will end up being constructed on the same properties. And the one here in the bottom panel on the left is just a sheet metal structure where multiple families will live on the same property. Unfortunately, we're not able to study the informal settlements, which are in the surrounding area around the smelter, and these are everywhere in South Africa, of course. These are much more transient, and so we don't really have the ability to determine the actual number of people living in these, and it fluctuates pretty dramatically over time. And there's also considerable challenges with safety and security for our workers. So over the 4 years of manganese exposure measurements, we were able to develop an exposure model that reflects the exposures that were occurring in the last 4 years in Meyerton. And this in the figure on your left, you can see in the warm red colors are the higher exposed areas. And as you see, we have an exposure gradient with New Sakelo having the highest exposure, Noldik second highest, and then Old Sakelo the lowest exposure within this region. And then this is graphically depicted with a box and whiskers plot on the right. Now the problem with this is that we started this study during the recession, the international recession, and the smelter at its peak was running up to 7 furnaces. During the time we did this study, the smelter was running 1 furnace. So the exposure levels that we were measuring, although higher than many communities in the world, probably likely are an underestimate of the exposures that were occurring over the last 30 years. And unfortunately, we've never been able to get actual production data from the smelter to be able to calculate an estimate of the different exposures over time. Ultimately, our goal is to look at the relationship though between our health outcomes and cumulative manganese exposure. So we are working on these data right now to come up with a more precise estimate of cumulative lifetime exposure. So our clinical assessments are conducted in 2 phases. The phase 1 visits are in the home, and here's a picture of one of my workers, Pila, interviewing one of our research participants to take detailed health history, residential history, and then we geocode the residents. We also collect toenails and fingernails to obtain biologic measurements from each research participant. And then in phase 2, research participants are asked to come to a central location in each community, and we do the health assessments in the community, including all the measurements, the health assessments that I discussed in in a previous slide. And here on the bottom right, I'm administering the UPDRS to one of the research participants, and you can see she actually has a baby on her back during the evaluation. The population that we're recruiting for this study is everybody who's 40 and older in the community, to focus on those people who are in the highest risk age range for Parkinsonian health outcomes. So we've had important cultural challenges along the way. The first was that English is not the primary language for many research participants. Most of our research participants speak an African language in the home, and we made every effort to try to administer the questionnaires in the African language that was closest to their home language. In most cases, it overlapped. There are 11 official languages in South Africa, but the region we're studying, primarily there were 4 languages that were spoken in the home. And I'll show you the challenge of that in a minute. The other cultural challenge we had was early on during the door-to-door recruitment, we actually had to halt recruitment temporarily because there were some residents of the community raised concerns that they thought our field workers might be witches. So it was not something that I'd encountered previously, but my team is very savvy in Meyerton, in Johannesburg. We worked with the local ward counselors to talk to reassure them, then reassure the community that our field workers were doing legitimate research. And we were able to overcome that challenge by just working with the leaders in the community. As many of you may know, that many of the people in the community rely on traditional African healers, like in this advertisement in the middle. And I was able to actually engage with one of our research participants was a traditional African healer, pictured on the right. She gave consent for me to show the picture, and I really enjoyed chatting with her. And she told me all about the stick that she has in her hand. This is her healing stick. She found it in a river that she said was left by her ancestors, and she uses it to heal various medical problems and even to help people with such things as finding their keys. A delightful lady. Her support and the support of the community really were key to us being successful in recruiting people for this study. So of course we had to validate some of the outcomes that we were using, and and I'll just show briefly a paper that we published in 2020, where we validated the Unified Parkinson's Disease Rating Scale in this kind of a setting against the PDQ-39, a commonly used Parkinson's quality of life questionnaire, and found a nice linear association between the two. We found a nice association between the UPDRS and Parkinson's screening questionnaire answers, which is the screening questionnaire developed by Carly Tanner to screen for symptoms of Parkinson's disease, and a similar relationship between the PDQ-39 and the Parkinson's disease screening questionnaire. So this provides some evidence of the validity of using these tools as we're using them in an environmental setting. Of course, we also had to validate our oral questionnaires, or the written questionnaires, which we actually administered orally. And here's an example of the validation of the state trait anxiety inventory. So in the English version of the STAY, there are two factors. I mean, so there's basically it divides into two components. There's an anxiety state component, which reflects the current anxiety level for a research participant. And there's an anxiety trait factor, which reflects lifetime anxiety. But in the South African validated versions of this, and we validated this in multiple languages, we found three factors. We found that the positively worded questions, whether they were state or trait, were one factor. The negatively worded questions were another factor. And then interestingly, in the state, this is the current anxiety levels, the negatively worded questions were another factor. And that proves to be interesting in a minute. But it just reflects the fact that when you start to use these Western developed questionnaires in an African population, they don't always behave exactly like you might anticipate. The recruitment wasn't always simple, and I made several trips to South Africa that ended up with me not being able to go in the field. And protesting is common in South Africa. There on the left is a picture of a student protest in about the middle of our study, and the students were protesting over their fees. Concurrent with this, unfortunately, we're protesting the community that we were studying related to housing and poor housing conditions. And this, when there was rioting going on in the community, we could not go into the community. And you see two pictures of the results of the rioting that occurred on multiple occasions there. Rocks, big boulders were put in the middle of the road to stop cars from passing. And then in a remarkable engineering achievement, overnight, people from the community were able to dig this trench in the highway with no heavy equipment whatsoever. It was just done manually. But these sorts of challenges reflect the cultural challenges that we have to deal with and the safety challenges in doing some of this work. So despite that, we persisted, and we were able to, and we saw early on that it was going to be very important because of the high level of exposure in the community to bring in a lower exposed community. And so this slide just shows a comparison of household demographics between the two larger communities that we're studying, the Midvaal and then Mogali City, which is where Ithembeletu, our reference community, is. And as you'll see, these are largely comparable communities. They're both, the settlements we studied are the requirements to live in them are the same as for each community. So people are largely very similar in education level, as well as the level of people with no work whatsoever. There is slightly higher level of employment in the Ithembeletu settlement as compared to the Meyerton settlements. But that's the community at large. The specific research participants, this is a direct comparison. And I'll say that we were successful in recruiting over 832 people into the study, and we had of those 732 were participated in phase 2, and we have health outcome data on, and we had to exclude some individuals because of comorbidities that made some of the evaluations impossible to interpret. So you'll see the data for all participants in the middle, and then Meyerton, which is our exposed community, and then Ithembeletu, our reference community. And you'll see that largely the communities are quite similar, and the research participants are quite similar with respect to the demographics. The male, there's some greater portion of research participants in Meyerton that are male. Almost all the research participants identify as Black African ancestry. Education levels are fairly comparable. There is a greater use of alcohol and cigarettes in Meyerton, and we do adjust for those variables. And the age is actually slightly higher in Ithembeletu. Once again, we adjust for all of those variable covariates in our analysis. So Wendy Lamini from my lab has been doing many of these analyses, and just want to give you a snapshot in the last few minutes of some of the key findings so far. This work is actually in review right now, and in this study we compare the motor tests that we conducted between Meyerton and Ithembeletu, and we see that in our study that the UPDRS score is 6.3 points higher in Meyerton as compared to Ithembeletu, and that's largely driven by bradykinesia. Consistent with that, we do see significantly or nearly significantly higher longer times to perform the groove pegboard test, and but most notably in non-dominant hand. But then in using a computer-based kinematic testing platform, we were also able to show that research participants in Meyerton were much slower in performing finger tapping tests in that community as well. Just providing some validation or some verification of the findings in the UPDRS. Consistent with that, we also find that depression and anxiety severity scores are higher in the Meyerton region compared to Ithembeletu, and it's most notably for the Beck Depression Inventory. And once again, you'll see in the line here that says BDI, you'll see that the difference in mean scores with pretty much any adjustment is greater in Meyerton versus Ithembeletu. We see a somewhat higher anxiety state score, but minimally different trait score. And these scores basically just show, normalize the two to show that BDI is clearly more effective than the anxiety state and trait. And then finally, our health outcomes, if you look at the comparison between the communities, our cognitive control outcomes, or cognitive outcomes, I should say, demonstrate that the matrix reasoning, this is the nonverbal IQ performance, is poor in Meyerton. The go-no-go test is poor in Meyerton. The digit span performance is poor in Meyerton. So for the most part, all three of these health outcome variables seem to be worse in the Meyerton community versus Ithembeletu. And we're currently working on the exposure modeling, cumulative exposure modeling, to look at the association between cumulative manganese exposure and these outcomes as the stronger evidence of a causal relation. And I'll just tease with this one slide. So we did a sub-study where we did MRIs, and we were able to do MRIs in 60 research participants from the communities. And this is a graph from some of the early, the first half of that data. And we were actually able to show that the MRI signal intensity in the substantia nigra, the region of the brain associated with Parkinson's disease, is actually, there is actually a bit of evidence of a relationship between that signal intensity, the brightness, which reflects manganese deposition, and the severity of Parkinsonism. And this will be followed up with a new R01 later this year. So as with any study in a developing country, we felt it was really important that we provide something more than just doing research in the community. So we have had some focus on education. Jen Goldman from Rush and I have taught both at Vera in the Baragwanath Hospital in Johannesburg, as well as Badunsa near Pretoria. We've provided employment for numerous people in the community who've developed skills that they wouldn't have otherwise gained by doing this work. And we've really tried to focus on building capacity through training. So Wendy Lamini did a master's with me at WITS, and now she works in my lab in the United States. That's for the last two years as a data analyst. Julian is a graduate student working with me, and Jill on her PhD in Johannesburg, working with our manganese miners study. Guinyai is going to be joining us as soon as the travel restrictions lift. He's going to join us as a postdoc working on the smelter work study. And then Anna Marie is doing a master's at WITS on a child study in Meyerton. So where to next? And this really is relevant for potential collaborations with the International Parkinson's Disease Genetic Consortium. We are working in the dose response and hope to have that data completed hopefully later this year. I told you about the biomarker development. And we recognize that dissemination is really a challenge in a community like this. These are complicated findings that we have to explain to the community, and we will likely be seeking research funding to better understand how to provide information to the community and develop materials for that dissemination. But ultimately, we're interested in really making the community safer and to bring this full circle to this theme of environmental justice. And I think that path may be really long, unfortunately, because we have strong evidence from some of the monitoring data that even if the smelter shut down tomorrow, that there's so much manganese in the soil that resuspended dust is likely will contribute to adverse health outcomes for decades. And I'll just finish by saying that we are in conjunction with the smelter study, and in the miner study, we've started we're developing a new study in the Adjun cohort up in northern South Africa, where we will be looking at social determinants of health and Parkinsonism in their cohort. And this would be a fantastic opportunity potentially to collaborate with the International Parkinson's Disease Genetic Consortium, as we'll be doing population-based assessments of everybody in this in a subset of this cohort. And so finally, just to acknowledge, this is work that has been conducted by 2 teams in 2 continents. Got most of my lab here on the left. In modern times, we've had focus on meetings by Zoom, even in our lab, and that was yet another challenge on the path to our work, but we've made it work. We have a great diverse group in the lab that is doing cutting edge research in Parkinson's disease. And then my team on the right, I'm very proud of the work that we've done in doing some very difficult field work and pulling together a project literally from nothing. And we'll just end with a picture of Nelson Mandela, and I'd be happy to take any questions.
Thank you very much, Professor Racette. It was a very insightful talk, and I found it particularly amazing how you face so many challenges, with regards to facing subpoenas from industry, the security challenges, the riots, and the cultural challenges as well, and how you overcame that. So thank you for sharing your journey with us. If anyone has any questions, please leave them in the Q&A section. I have a few questions of my own for you, Professor Racette. I was actually wondering if you could elaborate further on the biomarker research and where you see that going, and the overall outcome you'd hope to see, whether that would be regular screening for people that might be exposed to manganese.
Right. So I didn't give a detailed talk on manganese, and so when I give a full talk on manganese, one of the challenges that I always explain is that we do not currently have a good biomarker of cumulative manganese exposure. Our studies demonstrate pretty strongly that cumulative exposure is the key, and that measuring an 8-hour time-weighted average of manganese on one day or 1 week does not really answer the question about what health effects are associated with manganese exposure. So there are, MRI is probably one of our better biomarkers, but it's impractical in most areas. I mean, with that said, we have done 60 MRIs as a pilot study, so that alone is pretty remarkable to be able to accomplish MRIs in a random sample of people from a cohort. But the hope with this, and this is very much this was a very high risk grant that was funded by NIH, is that a salivary biomarker, which is obviously very easy to obtain with staff with very little training, could be could be validated in this cohort where we've done all these detailed exposure modeling measurements, and that microRNAs happen to be a focus of my collaborators' labs, and so we decided to take a risk, look and see whether there were any microRNAs in human saliva that would be associated with the exposure or health outcomes. And as you can imagine, it would be a very low-cost way of monitoring exposure and potentially even outcomes. And I will say that this is really, take this with a huge grain of salt, but one of the microRNAs in our first look at this, we just got the data 2 weeks ago. One of the microRNAs that seems to be associated with the model exposure actually does seem to interact with alpha-synuclein. So it would be very interesting if we were able to tie this together at that level. And I say this with a huge grain of salt that may not pan out, but it just shows you how innovative and how quickly we could advance the field if that actually proved to be true.
Okay, thank you. That's very interesting. We have a question from Moroni Keji. The question is, what are the differences between the presentation of manganese toxicity and idiopathic Parkinson's?
Right. So that a lot of the work we've done has tried to address that, and I can tell you that in general, in people who are very highly exposed to manganese, the syndrome seems to be more symmetric, but the early literature from Rodier's time in Morocco, the syndrome that Rodier described does not seem to be the syndrome we see in modern times. So it's really in modern times, and I've examined over 4,000 people with manganese exposure, just to give you a sense of the scale here. And the syndrome really looks like relatively symmetric Parkinsonism, little dystonia, not much tremor. Now, we're interested in, does manganese cause Parkinson's ultimately? That's not the work we've done so far. We've been doing work that gets at whether manganese causes Parkinsonism, and it's pretty clear it probably does. That's pretty clear, and in a dose-dependent fashion. At what level is what we're trying to address now. But I think that when you look at the bulk of the work we're doing, and I didn't show a lot of the studies we've done, we think that manganese, it may be a risk factor for PD, but more importantly, it may actually be a disease modifier. It may affect the phenotype of the disease. And as anybody on the Zoom knows who sees people with Parkinson's disease, Parkinson's is a highly variable phenotype, and some of that variability we believe may be due to environmental exposures. And it may be one of these areas where the kind of work that my lab does could be helpful for the consortium as well.
Thank you. Okay, I have another question. In your opinion, what do you see as the most promising therapy out there that's being developed at the moment, targeting?
So Fatima, you cut out. I'm sorry. I missed part of what you said.
Sorry. Okay. Sorry. I'll repeat it. Can you hear me now?
I can hear you now. Great.
Okay. I was asking, in your opinion, what's the most promising therapy out there at the moment that's being developed targeting manganese? I think I've seen something about targeting D2 receptors, etc. So I just wanted to know your opinion.
You mean symptomatic or like for manganese toxicity? Is that what you're saying?
Yeah.
Yeah, so the answer is I'm not convinced that we have anything right now, and I think that that's actually a really important, that's an important point. I actually think the work we're doing points to the potential role for modulating neuroinflammation as a way of mitigating the exposures to manganese toxicity, or mitigating the risks associated with manganese exposure. Chelation has a long history of very equivocal results. Symptomatically, I've treated a fair number of people, and the literature would suggest that people with Parkinsonism due to manganese toxicity may not respond to dopamine. My experience has been people just haven't tried high enough doses, and that in many cases that the people who are symptomatic from manganese toxicity may actually respond to levodopa. But I'm particularly intrigued by some novel approaches that have been considered to try to reduce the downstream inflammatory effects of the metal. Once the metal gets into the brain, it's not entirely clear to me that you can do much about the metal that's in the brain, because it seems to induce an inflammatory cascade of events that leads to neurodegeneration.
Okay, thank you. I have another question, which is, do you have any pieces of advice for any member of the audience who's in the early stages of their research career? Any tips going into a similar field that you've been doing in Africa? Do you have any pieces of advice?
So I think that there's no substitute for mentoring. And just to give you an example, and the the sort of the richness of the research environment in sort of NIH funded labs like mine really does foster research careers like nothing else. So take Wendy as a good example, who's in my lab right now. So Wendy did really nice work at BITS, but she was very much constrained by the quality of the data we had, and just sort of the limitations of the environment around her. Even though BITS is a fantastic research institution, the resources that she found when she came to my lab allowed her to be highly productive. She's supported by data managers, research coordinators, by a team that helps her to be sort of maximally successful. I mean, she's on 5 or 6 papers already, and it's just a matter of, it's just scale. And I think that having a mentor who can help walk you through the process of how you develop an independent research career, I think is probably the most important decision you can make.
Okay, yeah, that's really true. Thank you very much. I don't see any further questions. So on that note, I'd like to thank you, Professor Racette, for joining us today and giving us such an insightful talk. And thank you everyone for joining us today. We have another talk on the end of February. So we have a talk every last Tuesday of every month. So thank you very much, Professor Racette, and see you. Thank you.
Thanks for your invite. Take care, everyone.